Hello, everyone. So I'm Glaucia Fragoso. I'm a researcher, previously at NTNU, but starting today at Sintef Ocean. Hooray for me! Yes, and today I'd like to give a talk about the research that I have been doing the last years, which is looking to the environmental interactions between phytoplankton blooms, bryozoan larvae abundance, and encrustation in a kelp farm of the mid-coast of Norway. It's a really long title. So this research is part of this Monetara project, which was funded by the Young Research Talent, the NRC. And the title is Autonomous Underwater Monitoring of Kelp Biomass, Growth, Health, and Biofouling using Optical Sensors. And it had two main work packages. The first one is to use underwater robots to place them in a kelp farm and to look into the image analysis and compare that to the real biomass that we take by hand, and to see if we can use those robots to kind of speed up our biomass estimation of kelp in a kelp farm. And the second one has more of this ecological aspect of it. We have a lot of problems in the kelp farms, which are bryozoan biofouling, and we want to understand what are the environmental factors, both biological and physical and chemical, that is related to the appearance of these encrusting organisms. So this is a little bit of the background. So I don't know if you guys are familiar with this bryozoan biofouling, but if you guys walk in any beach here in Norway, you see that the kelp has these white patches, and those are encrusting organisms, bryozoans. And they grow in a kelp farm around May, June, depending on the area, but here in Trøndelag is around May, June. And they are very bad for the quality of the kelp because they make the kelp more brittle. They kind of like they are very bad for the kelp when they want to do photosynthesis, so they kind of act as shading. So what happens with the farmers is that they want to go there in the farm and they want to harvest before the bryozoans they start to appear. We have two main species. One is Electropilosa in the left and Membranipora membranacea in the right. And these organisms, they have a complex life cycle where in May, June, when they appear in the kelp, they are kind of a benthic, they are sessile, they are fixed in the kelp. But before they get to that stage, they have a larval stage, a zooplankton larval stage in the water column, where they are microscopic. So we know that from the literature that has been published is that temperature is really related to the appearance of these bryozoans incrustation. But we know that in real life, temperature correlates with other environmental factors, which is, for instance, the stratification is when the water column is more steady, also salinity, and also their own food, the food of the bryozoan larvae, which is the phytoplankton blooms. And the phytoplankton, they vary a lot. You can find like bacteria size, which you call picoplankton, but they are also found in larger size. So I want to understand, to monitor the phytoplankton blooms and temperature and other factors, and try to understand what is actually starting this encrusting of the bryozoans. So what we did in some sampling from January to June of 2022-2023 in a kelp farm of Seaweed Solutions in Freya. We sampled monthly or like 2 times in a month. And we collected water samples for phytoplankton size structure, but also for nutrients, and net samples for these bryozoan larvae. We wanted to quantify them to see if they act as a proxy of when the bryozoa is going to start to appear in the kelp. We also placed a sensor there, and we looked into changes of temperature, chlorophyll, and turbidity every 10 minutes from February to June. And in these water samples, we also look into the size of phytoplankton, because as I said, the bryozoan larvae, they prefer a smaller size because they are also quite small. And they filter water, so they select for the smallest one. And we also wanted to digitalize the fouling in the kelp And the reason for that is because the kelp farmers, they go there, they collect the kelp, but it happens that it's difficult to see them because when they're starting to grow in the kelp, they are sort of microscopic, like less than 1 millimeter. So we thought that maybe we can get them, we can freeze them, and later we can sort of scan and digitalize and quantify the colonies of these bryozoans in the kelp. And we also measure the size of the larvae using this microscope. So here is the larvae. We measure their size. This is the microscope, and this is the scanner that we use, just like a simple scanner. So we look into the environmental factors. So here we have like wind. As you know, our winter is like heavily on wind, and that causes a lot of water mixing. So the phytoplankton blooms, they only start to appear when we have more of a calmer conditions, when the wind starts to phase out and we have more sunlight, and that's usually in April and May. In 2023, it started in April. You see like a big peak in the top right there. And then in 2022, we have like, instead of one big peak of a bloom, we had like a start of a bloom and then more mixing and winds and then a second peak. So it's very common that we see multiple peaks of the bloom. And the water, they are colder, and the blooms, they only start to occur when you have like the temperature rise, which is usually in April and May, and then it keeps like becoming warmer and warmer. We also look into the size of the plankton. I forgot to mention, but in the SAS farm, we sampled within the farm, but also a little bit further away. So we compared the two sites, and we saw that the plankton usually is quite the same in terms of size. But the white one, which is the proportion of the smallest fraction, they start to be more abundant in May. And that could also be an indication, because that's when the settling starts to occur, so that means that maybe there is more food for the bryozoan larvae during this period when they start to settle in the kelp. And here we have like the general drawdown of nutrients because of the phytoplankton bloom. And here is like one of the most interesting things that we found. We look into the abundance of the larvae, of the bryozoans. In the up there is 2022, and the lower is 2023. In blue is Electropilosa, the species, and in yellow is the Membranacea, Membranipora membranacea, and we saw that they occur like in two peaks in both years. So first is around the bloom in April, and then we had a second peak in June, July. And we saw that even though we have that peak, the fouling only starts like here in May. So this is when you start to see the colonies in the kelp. And you see that they start around in May, even though they are abundant in April. But why does that happen? And the one found that we have is that it's not much about the abundance of the kelp, but the size of the larvae. Because these larvae, these bryozoan larvae, they take some time to mature themselves. They are in the water column, they are eating the plankton, but once the temperature starts to increase, they increase in size and they become more mature. So that means that when they're more competent, we would say, when they settle in the kelp and they form the colony. And we also saw that October had more of fouling, the red-pink line, compared to the January ones, because they have a larger area as well. So the main point here is that instead of just looking to the quantity of the larvae as an indicator of when the bryozoan was going to start to grow in the kelp, we should also look into their size, because that means that they are more mature, they're more competent for metamorphosis and for settling. We also saw some other things varying, not just temperature, but we also saw that abundance of larvae occurred more in stratified low salinity waters, maybe because the water is more calm, so they sort of like swim more around and they disperse when you have like more of this stratified low salinity, which are more like surface layers, so it helps them with dispersal. And we saw that the early kelp deployment, they had higher numbers of bryozoans, higher recruitment, with rapid colony growth from May to June compared to the January ones. And temperature is definitely a main driver, but other factors like post-bloom food, like the size of the phytoplankton, the fact that Froya is very productive region with a lot of peaks of blooms, and which means that there is enough food availability for both the larvae, but also the kelp that is encrusted in the bryozoans that is encrusted in the kelp, and also hydrographic conditions like stable stratification in May and summer helps the larvae also to settle in the kelp. And definitely temperature is correlating with everything, like sudden increase of temperatures, we saw more the appearance, fast appearance of these encrusting bryozoans. I would like to thank a lot of my colleagues, research assistants, Maya and Natalie, and also the co-supervisors of the master students that we had in Monitara. They are also from NTNU, SAS, and SINTAF, and also Oslomet. And also the people from NTNU, Seaweed Solutions, which opened the farm, and CTAF, and also to my 3 master students, which contributed to the results for this project. And we have now a paper that was submitted to aquaculture, is open, so you guys can take a look if you're curious. And you guys as well. Thank you very much.
Yeah, thank you, Glaucia. I think we have some time left for questions, if there's any questions in the audience. It's a bit difficult to see from up here, but I have some assistants. If not, we have prepared some questions. Maybe Clara, you could take those.
Thank you for your presentation. Based on your findings, is there any recommendation you could have for farmers if they are facing a year with a lot of storms, for instance, to harvest in a good time?
Yeah, so I think that the whole point of this research is like to really embrace monitoring. So we need to look into many environmental factors, and I would be really like I would pay more attention to the larvae, the size of the larvae that you find in the water column, because that can be an indication of when the settling is going to start to occur. And it would be very nice if kelp farmers, they could build like yearly, because you see that sometimes the fouling can start a little bit before, a little bit later, and that can help if you know the environment conditions, like we had an early bloom. We're starting to see that this is changing, the temperature is increasing rapidly. All of this will impact the quality of the kelp and how fast the settlement will help, and how fast the bryozoans is going to grow in the kelp. So the whole point is like to look into the whole monitoring. And I think with technology, things is going to start to become more real time, and because it can be a bit time consuming, but the hope is that maybe we can develop technologies that can make this job a bit quicker.
Yeah, thank you so much, Glaucia.
Thank you.